All right, so we got 14 participants, and I think it's about time to start. So welcome everybody in the next Petroleum Systems Analysis webinar. This time broadcast over in a new environment of Skype Business instead of GoToMeeting. And also, this is the first time we have a sponsor that actually helps cover the cost of the production of this webinar. It's the company called TIPS, which is basically a company of Andrew Pepper out of Houston. And you see his advertisement on the screen now, and you saw it in the announcements also. And I'm very grateful for that Andrew is doing this. Okay, our presenter today is Hartmut Jaeger out of Heidelberg. Hartmut is an organic petrologist. And I have just lost his profile here. Okay, I have it. Hartmut works as a principal geologist for hydrocarbon exploration in Georesources Transfer Center, which I understand is somehow associated with university, some sort of spin-off company. Is it correct?
Yes,
yeah.
Slightly associated.
Yeah. And we met a few times at the conferences, and I realized Hartmut would be a great guy to give us some introduction to petrography of vitrinite and the way it can be analyzed and other maturity indicators. So I will now give the presentation to Hartmut, and Hartmut, you can start.
Okay, I will take over. Good morning, everybody. At least here in Germany, it's a pretty early morning still, and we will start. I just have to take over the control of my screen. So I hope everyone can see now the starting slide. Vitrinite on trial. Is there anything better in organic maturation? So when I started with organic petrology and hydrocarbon system analysis, I was used to that vitrinite reflectance is one of the major tools in organic maturation and analysis, and that is a pretty valuable tool to work with. So let's see. Okay. So that's what you find in some books. I put some references here. Vitrinite reflectance, the major maturity parameter, accurately establishes the effective maximum paleo temperature and its duration at any stage in geological time. So this confirms that vitrinite reflectance is not just a tool, it's a very important and reliable tool. It's from the book in 1994, Vitrinite reflectance as a maturity parameter. And even in Wikipedia, for everyone, it's said that the study of vitrinite reflectance is a key method for identifying the maximum temperature history of sediments in sedimentary basins. And with this, it's also a key method for identifying temperatures in hydrocarbon systems, because they are controlled by the thermal history of the particular basins. But being in a discussion at the PETEX conference in 2014 in London, I was very much surprised hearing a lot of very critical discussions on the value of vitrinite reflectance. And one speaker stood up and said something similar than to the reference to this reference I wrote up here. Do you really believe in vitrinite reflectance analysis? We all know the weakness and the unreliability of vitrinite reflectance analysis from many, many studies. I was really very surprised to hear such a critical and negative statement on vitrinite reflectance, because in my opinion, vitrinite reflectance was a very strong and still is a very strong tool in maturation analysis. So the question came up, what is wrong with vitrinite reflectance? What are the problems? What is this that caused these problems and these mistakes in so many studies? And is there anything better in organic maturation analysis than vitrinite reflectance to change to? So this will be the questions the talk is focused around, to check the vitrinite reflectance. What is vitrinite reflectance? what are the benefits, what are the limitations, and also what other organic maturation parameter tools do we have, and are they really better than vitrinite reflectance or not? If you want to follow the talk, the headline here, you can always see where we are. Introduction at the moment, then we will get into vitrinite. What is vitrinite? Basics about vitrinite reflectance, methods of vitrinite reflectance, benefits and limitations, other maturation parameters, and then I will show you some case studies where vitrinite reflectance could do a major input on the interpretation of the project in the end. So let's go on with the question, what is vitrinite? I'm not sure how much you know about vitrinite, what your personal backgrounds are, so I just start from scratch. Vitrinite is the most common component of coals and of terrestrial derived sedimentary organic matter, what we normally call kerogen. So here it's already says terrestrial derived sedimentary organic matter. It's terrestrial in origin. It is derived from woody material of higher land plants. And this will have some consequences that we will see a little bit later in the talk on the availability of vitrinite. Chemically, it's composed mainly of cellulose and lignin. So these are highly complex molecules, cellulose on the left, lignin on the right. Polysaccharide is cellulose, lignin is a biopolymer. And you see here a sketch on plant cells with all this yellow to brownish stuff, which is mainly made of cellulosic material, and then these very thin layers in between that keep the plants very stable and upright is made of lignin, which is a very strong and resistant organic substance. That's the reason why plant fragments are that stable. It's not because of the cellulosic infill, but on the lignite that covers the cellulosic cells. Lignite is very resistant against many microorganisms. It's very slow breaking down in aerobic breakdown conditions. It's mainly degraded by fungi, and that's the reason why phytoclasts, so the particles of plants, are that stable and have such a good preservation potential in organic samples. So just to give you an idea, what is vitrinite? On the left, you see vitrinite particles in normal light. You can see very typical structures, always these very long, elongated ribs that we see in most of these components. And you can see cuticles here, typical plant structures from the inner part, from the wooden part of plants. And you can also see in this slide that we can have certainly different preservations, very well preserved material here and there, and less preserved material here, here, and here. So we usually have a mix of different preservational and also very often maturation stages of vitrinite in our kerogen in one sample. On the right side, we see the same vitrinite, not exactly the same particles on the left, but we see a typical range of vitrinite particles on the right in incident light microscope. So it's always gray, and it looks completely different because here on the right side, we don't see the normal surfaces of the fragments. These are all polished surfaces that we use to study vitrinite reflectance analysis. So we concentrate the organic matter, at least in our lab. We don't work with polished rock samples or coal samples. We isolate the organic matter from the rock, concentrate them in a resin block, and then the surface gets polished. And on this polished surface, the vitrinite reflectance is measured. So this is vitrinite. Vitrinite reflectance is a pretty old tool. It was developed during the 1930s to 1940s for the determination of coal ranks, the maturation of coal in coal petrology. So it is in the end a tool that was developed for coal analysis. Since the 1960s, vitrinite reflectance is more and more applied for maturation analysis and hydrocarbon exploration, because we work with very similar organic matter particles than in coals, but there are some certain differences, which we will see in a few slides, make a difference in the method to analyze this. Vitrinite reflectance measures the percentage of incident light reflected from the polished surface of vitrinite particles. So we always need a polished surface, and this polished surface has to be absolutely planar to have the precise conditions to analyze the reflectance of light accurately and to get proper vitrinite reflectance data. You can measure vitrinite reflectance on the polished surface of rock or coal samples. So you just take the coal or rock and polish and cut it and polish the surface and measure this on the surface. Like here on the right hand side, this is a rock sample with very few vitrinite particles, inertinite here, some vitrinite over here and over here. And a lot of this gray stuff is not vitrinite, but are different minerals. Or you can analyze. Hello? Okay. Can you hear me?
Yes.
Okay. Can I go on? Okay. So on the left side, you see a resin block with kerosene concentrate, and that's the second method you can use to concentrate the organic matter in a resin block and just polish the surface of this resin block and analyze the vitrinite here on this polished surface. The big advantage, in my opinion, is that you can concentrate all the organic matter from your sample and you get much more vitrinite on your surface to analyze than in the rock, where you just have the few fragments that you have in your rock. And if you think about, let's say, even a good high TOC rock with about 5, 6, 7% TOC, this means 5, 6, 7, 8% of this surface will be made of organic matter, but surely not all vitrinite. So vitrinite will be even less. So in the end, you have maybe 1, 2, 3% of your whole surface where you have vitrinite that you can analyze. Here in the concentrate, you have much more. You can concentrate the organic matter completely, and you get much more vitrinite particles here, like you see here, this big one, this one here, also this one here. So it's just a better way to have a concentrate, to have more samples to analyze. And that's the reason why I guess still more and more people change from polished rock and coal samples to resin blocks, but still there is quite a big community working with rock samples. And again, when I talk to companies, when I talk to people, it's always a question, why do you use resin blocks? Is this really equivalent to working with polished rock slabs? And in my opinion, it's not only equivalent, it's even much better, as I explained before. So one point that we have to keep in mind is the anisotropy of vitrinite. So the reflectance is not only differing from maturation, but it's also differing according to the different axes of our vitrinite. So in vitrinite, we have a long axis and we have a small axis. And this means like all minerals and things like that have an optical anisotropy, it always depends in what direction you analyze your vitrinite particle. If you get the maximum value along the long axis, very often marked as Rm with a big M, or today more and more Rmax, means maximum reflectance, or if you have the minimum, along the short axis, the Rm or Rmin, which means minimum reflectance. So the difference between R max and R min depends on two factors. The one factor is how you cut or how you orientate your analyzed surface, which you see here in this diagram. So you have three different surface orientations. Here you have the maximum difference between R max and R min. Here you have a sort of intermediate, somewhere in between the maximum difference, and here you have a cut where R max and R min is almost the same. So this is one point that you have to keep in mind, and the other point is this difference between R max and R min increases generally towards higher maturation. So on maturation levels above 1.5 to 2%, it gets more and more important because the difference between R max and R min becomes more and more obvious. The analysis of R max and R min is mainly applied in cold petrography because you have large vitrinite fragments, and you can, just by rotation, you can quantify the reflectance and exactly measure R max and R min. So in cold petrography, it's a commonly used analysis. But in dispersed kerogen, and this is what we mainly have if we work with shales, if we work with hydrocarbon source rocks and hydrocarbon systems, in dispersed kerogen, when you isolate your organic matter and make a vitrinite block, you see a typical block surface here, you have a chaotic orientation of the vitrinite. It's not layered, it's not in one main direction like in a coal, but it's a totally chaotic orientation. So the analysis of R max and R min is more or less impossible. You can only measure pseudo R max and R min, means the maximum and minimum values of this chaotic mix. But you don't know exactly if this is really the real R max or the real R min, and therefore you cannot do any exact correlation with the real R max and R min values. So if you have measured R max and R min in a coal and you have perfect correlation with the coal and the maturation, it's impossible to make an exact correlation to the R max and R min in dispersed kerogen, because you never know if your pseudo R max and R min is really the same, the real R max and R min, like in the coals analyzed. So in the vitrinite reflectance analysis of dispersed kerogen, it is normally analyzed as the statistical mean of all vitrinite reflectance measurements. And it's, in previous time, it was often called RR or RM, random or mean reflectance. Today, it's mostly called RO. It's commonly analyzed with oil immersion at 500 times magnification. And because of the oil, it's RO. So vitrinite of dispersed kerogen is commonly presented as mean RO, based on 50 to 100 vitrinite particles per sample. This is our basic principle with the 50 to 100 vitrinite particles per sample. If you look in literature and in the internet, just when I checked several pages preparing this presentation, I realized that quite a number of people talk about at least 30 vitrinite particles. In my opinion, the statistics won't be good enough if you only have 30 particles for your analysis. But that's the major difference between the classical application in cold petrology with analyzing R max and R min and the application in dispersed kerogen like here, where you have the total chaotic mix and you just work with statistical mean values. So the technology, how is vitrinite reflectance analysis done? It's often, still today, often done with a photometer analysis. You have a microscope with a photometer unit on top, it's in between. Here you have an aperture with a field, measuring field here given by the aperture, and then you analyze your polished surface, but this means it's not just one point that you measure, it's a whole area that is measured. So in this defined area, the total reflectance of this area is analyzed, and the area depends on the diameter of the aperture, which is often something between 50 to 100 microns. So this method works very well if you have large sized vitrinite particles, like typical in coals. This picture here, it's a vitrinite in polished coal. So you have a huge vitrinite particle, another one here. And if you measure with this measure field, you will have a very homogeneous value. So for coals, where this method was developed for, no problem with the method. But if you go back to the dispersed kerogen, the picture that I showed you before, you have the same measuring field here. We have relatively small vitrinite particles. And if you now measure the reflectance of this whole field here, we get a mixed reflectance from different particles, like the vitrinite particles, this bit of the inertinite, a bit of pyrite, and the dark background. So it's not only the vitrinite reflectance that is given with this measurement, it's a whole mix out of these different materials in the measuring field. So if we use this method in dispersed kerogen, we need a correction factor. And so a lot of people develop their own individual correction factors. For me, the problem is that every measurement you do has a new mix. So this sample here has exactly this mix. If you go to the next particles, you get a very different mix. Maybe more pyrite, maybe more inertinite, more light-colored stuff, maybe more dark background, but it won't be surely not the same mix as we see here in this measuring field. So in the end, each measurement would need an individual correction factor. So how can this work? Is it really possible to correct all these totally different mixes of measuring fields with a handful or one or whatever correction factors? In the end, you would have to correct every measurement you do with an individual factor. Dispersed kerogen is common in shale samples, where we isolate the organic matter from the rocks, which are commonly used in hydrocarbon exploration. So in difference to coal petrology, where we have coals with big particles, in hydrocarbon exploration, we mainly work with shales, and we mainly work with dispersed kerogen like this one here. So there is a need for new analytical methods for vitrinite reflectance in hydrocarbon exploration to get more reliable vitrinite reflectance measurements, and which are based on the reflectance of the vitrinite particles only, and not on something like a whole measuring field like here. So how can we solve this? There are different options. This is an option that was developed by a friend of mine, also working with organic matter and organic maturation, and a friend of him who is a computer specialist and developed image analysis software modification to analyze digital images of vitrinite reflectance. So this is a modern approach. We no longer analyze directly the reflectance of the light from the vitrinite in the photometer on top of the microscope. We take digital images of vitrinite in a black and white mode. So we get these grayscale pictures and analyze the reflectance by grayscale analysis from this picture. So it's based on grayscale analysis of these digital images of Vitrinite and the calibration standard. Here on the left is a calibration standard. This is a sample that we want to analyze, are taken from an incident light microscope under the same conditions. And then the main work for the program is to transfer the grayscales into Vitrinite reflectance values, like 255 grays values. And with the calibration standards, we calibrate the software. You see the yellow points, which are the standards that calibrate the system. And then if you have calibrated the system, we can just use the different grayscales to get exact Vitrinite reflectance values. This enables us to do precise Vitrinite reflectance analysis on small Vitrinite grains, like here. These grains are only about 10 to 20 microns big. And we can do it even down to pixel size, to only a few microns, depending on your image resolution. So we no longer measure an area, we just measure a point. And this point depends on the pixel size, on the image resolution of your system. Besides this advantage that you no longer work with measuring fields, but can do point measurements, this method also has the benefit that you have an optical control of your analyzed particles. So you can check the type of organic matter and the preservation. And even if you have done all your measurements and you start with the interpretation of your data and you realize that something seems a bit strange in some samples, you can always go back to the exact sample pictures, to the exact fragments that you analyzed, and can check what's going on. And like you see here, you see different values from 1.08 to 1.72. So depending on this is all in one sample, in one maturation. So depending on the preservation, you see here a very fluffy altered Vitrinite, which has significantly lower reflectance than these well preserved Vitrinite grains up here. So this gives us an additional benefit with this method to have an optical control of the particles that we analyze, which is much better than the analysis with the photometer. So this is a technique we use. So what are the benefits of Vitrinite reflectance analysis? Why is this strong tool that is used in many studies, is taken as reference in many projects? And let's see. A very big advantage is that you can do precise measurements and have a lot of well-established correlations. So this is a big point for Vitrinite reflectance. You can do very precise analysis of maturation. You can measure exactly how much you want one number behind the dot, or 2 or 3 or 4 or whatever you need. So it's up to you how precise you want to analyze. It is a highly reliable and fully repeatable and verifiable analytical method. So if you have your resin blocks and you have done your measurements, you can repeat these measurements again and again. You can give it to someone else who want to test if he gets the same values. And as long as you don't start to polish the surface, if you get scratches on your surface and you have to repolish it, then of course you might get different particles, which might give some minor differences in the general reflectance data. But as long as you don't start to repolish the surfaces, the surface, you always have the same particles. You can look with a microscope for this exactly the same. So I measured this the last time, I measured this particle this time, and again. So you always can control the quality of these data. Another big advantage with Vitrinite reflectance is that it has a lot of well-established correlations with several other maturation parameters. As we see in this figure here, there are certain microscopic parameters and also chemical and mineralogical parameters which are correlated to vitrinite reflectance. TAI, thermal alteration index, which is an index of polynomorph colors. Conodont alteration index, which is focused on conodont colors. Then the whole fluorescence color analysis is correlated to vitrinite reflectance. Also, bitumen reflectance, solid bitumen in samples, is correlated to vitrinite reflectance. And also chemical parameters like Rockeval Tmax data are correlated to vitrinite reflectance. Electrocrystallinity and also the illite ratio in mixed layered illite smectite clay minerals is roughly correlated to vitrinite reflectance. And we also have well-established correlation with the major stages of hydrocarbon generation, what you see here. So the lower immature interval, where you just have biogenic methane and immature heavy oil generation, the oil window, which is more or less given by the vitrinite reflectance from 0.5 to 1.35, wet gas to and condensate field, and the dry gas field up here. So vitrinite reflectance is in a way a sort of calibration reference in organic maturation studies. And very often it is used as a sort of reference correlating one maturation parameter with others. So it always gets correlated to vitrinite reflectance, and then the other standard gets correlated to vitrinite reflectance, and so we get a correlation between different organic maturation parameters. So this supports that our vitrinite reflectance is important and seems to be reliable, because if not, all these correlations would be rubbish and wouldn't be done, because why should we waste so much time for rubbish? So there must be something on vitrinite reflectance which is really reliable and important to use it as this strong calibration tool. The other point, which is a big benefit on vitrinite reflectance, we can calculate paleo-temperatures. And this is quite good, much better than with a lot of other maturation tools that we have really well-proven empiric equations where we can highly precise calculate the exact paleo temperatures, the maximum paleo temperatures the rock has suffered, and from vitrinite reflectance. And you see that there are different equations. Of course, there's not only one equation. Different people work on this, and here on the right you see how this works. So there are a lot of data produced in several studies, which are then plotted together, the temperature versus the reflectance, and you see how you develop your reflectance correlations. One of the first was the equation given by Price in '83, followed by Barker and Pavlievich in '86, which is a very commonly used equation for maximum temperature calculation from vitrinite. And here, this Rm doesn't mean Rmin, but mean R, so statistical mean of the reflectance data. Then a very simple equation by Barker in 1988, which is also very often used for temperature calculations, and then the equations by Barker and Goldstein, which are correlating the mean reflectance with homogenization temperatures from fluid inclusions to access the maximum temperatures. So this is not a direct correlation from vitrinite reflectance to paleo temperatures, but it's a correlation or calculation via fluid inclusion temperatures to T peak, so the maximum paleo temperature. Of course, if you see the different equations, it would be, really wonderful if they would all come to the same results. Of course, they do not. The different equations lead to slightly different results because they are all a sort of description of this correlation, which is not exactly working in the well for all the particles. So depending on the data clouds the different people used, they get slightly different equations, and these differences in the results increase with maturation. So between the extreme equations, between the equation giving the lowest and the highest paleo temperatures, we have about, or slightly less than 5 centigrades at the basal oil window, but about 25 centigrades in the upper gas window, and over 30 degrees if you go up to the over mature area. So this shows these paleo temperatures cannot be just taken like a fixed temperature. They have always a certain variation, and so it's not a fixed temperature nailed down to 1 centigrade. It's a terminal interval. The higher up we go in maturation, the bigger this interval gets. But this is compared to many other maturation parameters, and the realistic calculations that can be done from other maturation parameters. This is quite precise. Limitations of vitrinite reflectance analysis. This point up here, I like to work with vitrinite reflectance, and I'm sure that vitrinite reflectance is a very strong tool, but of course we have to face the reality, and every scientific method has its limitations, and vitrinite reflectance analysis is no exception. So also vitrinite reflectance has clear limitations, which we have to be aware of if we work with vitrinite to get around pitfalls and simple mistakes that we could do if you ignore this. The first major limitation is stratigraphy and depositional systems. So one major point is that is limiting vitrinite reflectance analysis is the availability of vitrinite. And the availability of vitrinite is limited by stratigraphy and depositional settings, what we call phases. Vitrinite, as I said just in the beginning, what is vitrinite? Vitrinite derived from higher land plants. These are all terrestrial plants. So this means two significant limitations. First, vitrinite is not available before the latest Silurian due to the evolution of the land plants. So we know that the first land plants start to develop during the middle to upper Silurian, but it's just in the latest Silurian and then much more in the early Devonian that we have land plants in high numbers to produce a lot of material, so we can have a certain amount of vitrinite in our samples to work with. So for all systems we want to analyze before the latest Silurian, vitrinite reflectance is not available. This is quite an important point for all the early Paleozoic shale systems which are under investigation during the last years regarding unconventional shale gas and shale oil. And the second point resulting from this fact that they are derived from higher land plants is vitrinite is available in terrestrial derived sediments, but not available in very distal, not depth marine, but deep marine basins, where we have very low terrestrial input, and most of this low terrestrial input is inertinite. So this means totally coalified black organic particles, which are not vitrinite and therefore cannot be used to analyze the reflectance and calculate paleo temperatures. So in these very distal deep marine basins, vitrinite is almost not available, and therefore vitrinite reflectance is not able to be done. The second is certain carbonate sediment systems. Quite a number of carbonate sediment systems are where we have no or almost no terrestrial input. So we also have no plant input and no vitrinite input, and therefore vitrinite reflectance cannot be used. And then all evaporitic sediments, where we typically have no terrestrial input, all these sediments are out of use for vitrinite reflectance. So these two are very clearly limiting factors. Apart from these, vitrinite is very, very widespread, is available across almost the entire time scale, and is a very widespread and common material to work with. But there are more limitations. So if we're in an environment where we have vitrinite, this doesn't mean that everything is fine and can be done without problems. This means that we have to check the vitrinite and identify if the vitrinite that we have in our samples is really valuable and reliable to work with for vitrinite reflectance. So the first point is vitrinite preservation. As I showed you here, these are typical preservations from well preserved to very poor preserved, strongly altered, degraded, and the same we see here on a polished surface in reflected light, very well preserved, sharp-edged vitrinite particle starting to get rotten and fluffy, and then we have a totally fluffy particle here. And just by these grayscales, these are particles from one sample. And if you see the grayscales, they get darker from well preserved to badly preserved, poorly preserved. So the increasing vitrinite alteration, degradation in this direction is corresponding to decreasing vitrinite reflectance, because the higher the reflectance is, the lighter are the particles. The darker the particles, the lower the reflectance is. And also, it's not only that the reflectance is lower, is abnormal low, it's also less reliable, because you don't have a clearly surface out of which shows just more or less one gray scale. You have a mix of different types of gray in this very fluffy component here. So the preservation of vitrinite has a strong effect on vitrinite reflectance. We have to, there we have two types of alteration, degradation. The first one is the primary alteration, which happened during transport and deposition. As we know, all these particles come from land plants, and a lot of vitrinite that we work with are in marine environments, shallow marine environments, shelf environments. So they have quite a transport to get into these systems. And during the transport and also during the deposition in these sedimentary systems, the organic matter starts to get altered by chemical processes, oxidation, but also a lot by microbial attacks, which start to degrade the organic matter. And following this primary alteration, we have a secondary alteration, which is related to the maturation and hydrocarbon generation. So if the material is stored in the sediments, if the system gets closed, so no more alteration can happen, and we have something preserved like this. During ongoing subsidence, during ongoing heating and maturation, this particle at one stage starts to produce hydrocarbons. This one here, because plant fragment, kerogen type 3, it's relatively late starting to produce hydrocarbons. Mainly, it's gas producing, so you need quite some temperature, but if we get into this maturation level, this particle, well-preserved particle, starts to get decomposed to produce hydrocarbons, because the hydrocarbons come out of this, so this well-preserved particle has to get decomposed. to generate hydrocarbons. And this is a secondary alteration that leads to very often badly preserved organic matter in highly productive source rocks. And up to from a certain limit, the particles are so badly preserved that really reliable vitrinite reflectance analysis is no longer possible. So if we have a sample mixed, like here, this is one sample. As I said before, we have well-preserved material, we have poorly preserved material. So for the accurate maturation analysis, these altered and degraded vitrinite clasts, like these ones here, have to be excluded from the well-preserved vitrinite, and the maturation analysis should be based only on these well-preserved vitrinite particles, not on this rotten stuff over here. The other point is, okay, let's go up again, it's too fast. The other point is the vitrinite identification, and this is not so easy as it might seem. So we have 3 different particles here. The left one, again, this wonderful vitrinite particle. Then we have something which in a way looks similar to types of vitrinite particles that I showed you on this third slide in the very beginning, but is in fact a particle of graptolite, something completely different. And we have a chitinozoa, an almost complete chitinozoa here, which also looks somehow similar to vitrinite, but is something completely different. So these 2 things here, graptolite and chitinozoans, are made of a totally different material. They are mostly made of different types of chitin, while vitrinite is mainly is mostly made of cellulose. So on the polished surface, in reflected light, the identification or the separation of these different types of organic particles can be quite complicated. It's also a question of preservation of the particles. And graptolites, chitinozoa, some other zooclasts, and also sometimes amorphous organic material can look very similar to vitrinite. So this needs some experience and needs careful investigation to make sure that we only analyze vitrinite and not other organic matter particles that we have in our sample. It seems to be obvious that you only have to work with vitrinite and seems to be simple, but in fact it's not always that simple to really separate vitrinite from other types of organic matter. And if we mix up vitrinite with other types, then we get a certain error in our data. And then another important point after preservation and identification is vitrinite recycling. Recycled organic matter is common in most kerogens, analog to recycled minerals and the host sediments. So as I said before, vitrinite is a part of terrestrial land plants. So it's not deposited in place just where it is produced. It has a lot of recycling, a lot of sediment transport, deposition, mobilization again, another deposition and recycling. And also if these sediments get sourced by the erosion of old sediments, which might be high mature, we can get high mature organic matter recycled in low mature in situ sediments mixed together in one sample. So the recycled kerogen can differ significantly in maturation, and not between samples, but for each sample. So you have a less mature part of organic matter in your sample and a higher mature part of your sample. And that's what you see here in these figures. These are 2 samples from a study in northern Germany, which I will show you a bit more details later on when we are getting to the case studies. And we see here the 2 samples. The red one is a high mature recycled organic matter. The green one is a less mature in situ organic matter. And we see here, this sample is dominated by highly mature recycled vitrinite. So the mean reflectance of the total vitrinite assemblage, if we measure everything, is much higher than the mean reflectance of this in situ part of the vitrinite assemblage. You see here a big difference. In total, the mean reflectance is 2.02%, and the in situ material is only 1.18. So this means the in situ material is upper oil window, while the total amount is upper gas window. So this makes really a difference. And here in this case, we have only a very few recycled particles of vitrinite in the sample, mainly made of in situ material. So we see a relatively small difference. The mean reflectance of the total assemblage is relatively similar to the mean reflectance of the in situ assemblage. The in situ assemblage is 1.03, and the total assemblage is 1.19. So this means, yeah, something in the upper oil window, both values. So the mixture, the ratio of the highly mature recycled and the less mature in situ organic matter strongly controls the maturation of the total vitrinite reflectance or vitrinite population. And this has to be always taken in mind, that the vitrinites that we measure in one sample are not all in situ and are not all on one maturation level. They can strongly differ. And the total maturation is controlled by the ratio of the in situ versus the recycled vitrinite reflectance and the difference in maturation. It's not only the number of recycled versus in situ, it's also the maturation of the recycled part, which affects the total maturation. If you have less amounts of recycled material, but very high mature, then we get the same effect like with a lot of recycled material with less maturation. So these both influence the total maturation of our sample. Analyzing total vitrinite populations can lead to strongly misleading results and interpretations, and this is something we will see in the case studies later on. For accurate maturation analysis, recycled vitrinite has to be excluded from analysis. Only the in situ vitrinite gives the maturation of the studied basin. All this recycled part tells you something about previous hydrothermal system, polythermal systems, about previous subsidence and of basins that might be no longer existing because they are completely eroded. The only part of the vitrinite that tells you something about the thermal environment of your studied basin is this in situ part here in green. The same is true for caved vitrinite. When we work with cuttings, we can have a certain impact of caved material and therefore also caved vitrinite. But then the caved vitrinite is mostly less mature. So we have the less mature caved population and the higher mature in situ population. So far to the vitrinite reflectance, what it is, how it works, and what are the benefits and what are the limitations are. So we see it seems to be a good tool where we can work quite good with it, but it also has certain limitations that has to be recognized and taken care in our analysis. So now I want to have a short look on a number of other maturation parameters. It's just a really short look on each of these parameters. We could do another complete talk, but I just want to have a short look to see, so how do these parameters work? What are the benefits? What are the limitations? Are they somehow better or worse than vitrinite reflectance? So the first thing I want to look on is reflectance of non-Vitrinite particles. Particularly in the early Paleozoic shale systems, this becomes more and more used, because all organic matter shows reflectance of light. So we can use all organic particles and analyze the reflectance of light. And for the reflectance of non-Vitrinite organic matter, commonly graptolites, chitinozones, and scolecodons are used, which are made of a completely different organic matter than vitrinite, but also solid bitumen is used, vitrinite-like particles, which are not clearly known what it is, but look like vitrinite, and also unidentified organic matter, which is just organic matter and no one really knows what it is. For all these different types of organic matter, vitrinite is used for calibration. And this looks here in a work from Bertrand in 1987. It seems to be that the whole issue is solved. So we have telenite, which is a type of vitrinite. So this is our vitrinite here. And then we have the chitinozones, graptolites, scolecodons, spores. So, and they are already all correlated. So what is the point about this? In fact, also in this paper from Bertrand, this calibration is only based on a very few samples in this study. And the calibrations that we have generally from vitrinite to other reflectants of organic matter are very, very few studies and always very few samples. So we are far away from a continuous correlation from low mature to high mature. It's also very limited in maturation rank. It's more or less local studies, and we have certain points that we know how to correlate, but not the complete system. With this correlation of non-vitrinite reflectance of organic matter, also another term gets more and more used, the so-called vitrinite reflectance equivalent. This vitrinite reflectance equivalent is based on the correlation of non-vitrinite particle reflectance to vitrinite reflectance, but also used for chemical parameters like Tmax or illite crystallinity versus vitrinite reflectance. And particularly in these early Paleozoic shale systems, very often these vitrinite reflectance equivalents come up. But the problem is very often it's not really known what the organic matter is that we used to work with these vitrinite reflectance equivalents. So the origin of the organic matter is often not clearly known, and it's also sometimes pretty difficult to distinguish the different types of organic particles on the polished surface of the sample. Until today, there are only very few studies with few samples from limited maturation intervals. We still have not a single continuous study across a wide range of maturation for correlation between vitrinite and other organic matter particles. Therefore, we also don't know exactly how the maturation-related changes of these non-vitrinite organic particles work, if they show similar changing trends like vitrinite, or if they show a different trend. And then also, like in the vitrinite reflectance before, we have to take care on the effect of recycling and alteration of this non-vitrinite organic matter. Another method you can use is the optical analysis of organic matter in polynomorph color indices. So we see here vitrinite reflectance, and these are the two major systems that are used for color analysis: the thermal alteration index, the older system, and the spore color index, the newer system. And we see that we can, based on the color of polynomorphs, not only spores, but the same works with acrotax in the marine environment. So we can make a system based on the colors and identify the different levels of maturation, and also roughly correlate this to the main stages in hydrocarbon generation. It's a numerical scale with a maximum 10-point scale for the spore color index from translucent to black. It gives a fast overview on general maturation levels. It also is good to identify altered and recycled organic matter, because in the slides we look on these polynomorphs, we see the color distributions and can very rapidly understand if there is recycling in this assemblage or not. And we get a good estimate of hydrocarbon generation levels, a good first estimate. The normal light microscope colors can be combined with fluorescence light. So here we have the fluorescence of alginate and different maturation levels from greenish to light yellow, light bright yellow to red in the end. But fluorescence only works during the oil window. If you get into the gas window, the fluorescence of organic matter is gone. So for the oil window, fluorescence colors is good to use as a support for the normal light color scheme. The limitations are clear. The color analysis is subjective for each investigator. So what exactly is SCI 4 or 5? You see this in this color, I see it in this color. So 3 investigators might come up to 4 slightly different interpretations. It's based on maximum 10 levels, on even less levels here in the thermal alteration index. Therefore, we only get a rough estimate of Parley temperatures, not as precise as from Vitrinite reflectance. And again, we have the effect of alteration and recycling of polynomorphs that also has to be taken into account here in the architects. But because we work with a microscope, we see the organic matter that we work with, we can identify this alteration and recycling very well. Coming from the optical methods to the chemical analysis of organic matter, one of the major parameters used from Rockwell analysis for maturation is Tmax. Tmax is a temperature at the maximum rate of hydrocarbon generation, the so-called S2 peak up here. So this is the Tmax. It depends on the kerogen maturation, but also on the kerogen type, which is a sort of limitation, because if you don't know exactly the type, we might misinterpret this Tmax value. It's also like Vitrinite reflectance, a highly precise analysis of maturation, fully repeatable and verifiable. And it's also well established with a lot of other maturation parameter and stages of hydrocarbon generation. But like all chemical analysis, it's based on bulk rock analysis. So we analyze the whole mix of organic matter that is present in this one rock sample. So we get a mixed chemical signal from the different parts of the kerogen, the different types of preservation, the different types of chemistry depending on the different particles, but also the different types of maturation from the different parts of the kerogen. And in chemical analysis, this mixture is impossible to observe. So we cannot observe the recycling. If there is recycling of high mature kerogen in this sample, that can change the Tmax, and we also cannot identify if there is alteration, degradation of the kerogen, because we just measure the whole rock completely. But both the recycling and alteration has a significant influence on the chemistry and the kerogen interpretation based on Tmax. And we see it here. We see a very nice profile, as it should be, S1 and S2. But sometimes even in the pyrolysis diagrams, we see that there is something very strange. S1 here, S2, this is the real S2, this little hill here over here. Or here, even worse, we have the S1 peak, and then we have a double peak. So it's S2 here or S2 here, changing the Tmax completely, changing the maturation interpretation completely. And I can tell you these strange pyrolysis diagrams are not related to any contamination and artificial problems of the samples. This is purely related to strange mixes of kerosene in these samples that we afterwards analyzed in the optical kerosene analysis. So this sets a clear limitation to Tmax analysis, because we don't know exactly what is recycled, what is mixed, what is how the kerosene is composed that we analyze. Another parameter that is quite often used in studies is the production index, PI. And here we see as a rough sketch how it is used. Immature below 0.1, oil window 0.1 to 0.3, above oil window, so gas and overmature above 0.3. And the production index is the ratio of already generated hydrocarbons versus the total amount of available hydrocarbons. It's a sort of indicator on hydrocarbon generation, if it already started. This corresponds to the maturation, and therefore it's used as an indirect maturation tool. But the production index, of course, is influenced by several other factors than the maturation. And therefore, there's a high uncertainty in PI used for maturation analysis. And I just showed two figures here from a study in the Mesozoic of South America, where we see the data points here. And according to the Tmax classification, maturation scheme that I just showed you the slide before, this would be immature. Here we have the oil window, mixed zone, gas window, and from the system for the production index, below 0.1, all this here would be immature, up to here, oil window, and all this above is over oil window, gas, and over mature. And in fact, so we see the data, how we plot the data here. All these data, which are marked in red fields, are has to be totally around, are the inconsistent data. These data are here, are immature according to Tmax, but mature oil and even gas mature according to the PI. So green are the inconsistent data and red are the, green are the consistent data and red are the inconsistent data. And the same we see here, a little bit changed, because here we have taken Vitrinite reflectance as one maturation parameter versus PI on this. And we see again, most of the samples show a mismatch between PI maturation interpretation and Vitrinite maturation interpretation. And the same is true for PI versus Tmax. So PI seems to be a quite difficult and unreliable maturation parameter. But it's still pretty often used also in discussions about projects and studies. Very often people come up with the PI values, so it's in a way relatively common to use as an additional maturation parameter. Then we have the next group of chemical analysis of the organic matter is biomarker analysis, so the full analysis of the chemical analysis of the organic matter. So mainly during the maturation, large biomolecules get cracked into smaller biomolecules. The formation of aromatic rings is increased by the loss of hydrogen from the naphthalene, and we change the three-dimensional structure of the molecules. It means the RS isomer ratio, the clockwise and anti-clockwise construction between the different isomers of the molecules. And that's what we see here. This is an immature Hopan profile. This is a mature from the same sample. You see here, the SR value is as smaller or slightly smaller or the same like R. isomers, and here in the mature samples we have much more S isomers than R isomers, so the ratio has changed. And in general, we have a sort of shift from larger molecules to smaller molecules. So here in this part, the smaller molecules are increased, while the bigger ones are decreased. So that's these three things we can see here in these diagrams. And but the problem is, again, it is primary or is it secondary. That's something we cannot solve also with biomarker analysis, because we just work with bulk samples. And the other point is biomarker changes are also affected by lithophases, by the type of organic matter, by the heating rate, and other factors. So it's not only changed by the different maturations. So therefore, exact maturity for a given value of a biomarker parameter can change from basin to basin, or even in several intervals within one basin, if you get into a totally different lithophases system, or if you get a different type of organic matter in another interval. Specific biomarker parameters are applicable only over a specific maturation range until they are terminated. So, and the total concentrations of the biomarkers decrease with maturity. So the higher mature our samples are, so the less biomarkers we can isolate to use for analysis. And one point, very important point, is that biomarkers changes are generally nonlinear. That's what we see here. It's an example from the styrene isomerization from the ratio of R to S isomers, and we see this is the immature, this is a mature field, and it's not a continuous function that goes up to the mature interval. It's relatively similar in the immature system up to here, and then we have a huge jump into the high mature system here. Therefore, it is not really good to use for certain intermediate maturation analysis. Also here again, we have to take care of the effect of kerogen recycling and alteration. Another point is clay mineral analysis. So we can do chemical analysis of non-organic matter. Illite crystallinity is a quite common used parameter. Illite crystallinity changes during thermal overprint. We see the illite peak here, which gets narrower and higher in increasing maturation. And it's commonly measured with a Kubler index, which means the width of the half peak. And this is calibrated then again with this Vitrinite reflectance and other maturation parameters. But if we have a higher amount of mixed layers illite smectite clay minerals, which is quite common in many shales, also in Paleozoic and even early Paleozoic shales, this very huge white peak of the mixed layers illite smectites covers the illite peak, and illite crystallinity is no longer available. So in the case we have illite smectite mixed layer minerals, we have no chance to do illite crystallinity. Also, we have to take in account diagenetic alteration of primary clay minerals, which might give some strange changes in the chemistry. And one point again, also recycling of high mature clay minerals is a problem that we have to take care of in clay mineral studies. Another optical analytical method of non-organic matter is the Conodont alteration index, which was already mentioned before. Here we see the different color levels of conodont from light brown yellowish to dark and pale gray. And these conodont colors are linked to thermal overprint, darkening with increasing maturation, and then again lightening in the higher upper maturation. Paleo temperature calculation from the conodont alteration index is strongly time depending, This means it's less accurate due to the uncertainties of timing of your thermal overprint. And that's what we see here. Here we see the diagram. So for the conodont alteration level 2, depending on the duration of the heating, 10 years, 1,000 years, 1,000,000 years, we get totally different temperatures. The shorter the thermal event is, the higher the temperature has to be. But it makes a big difference if you expect your heating event to go for 100,000 years or 1,000,000 or 5,000,000 years, because then we get a totally different temperature, and that's the reason why we have these wide temperature ranges here. So even in the lowest level, there is a temperature given from below 50 to 80 degrees, second level 60 to 140, 110 to 200. So we see there's a wide temperature range, and it's very often not clearly to identify how long range this thermal event has been, and therefore what exact temperature we would get out of the conodont alteration index. One more optical analysis of non-organic matter fluid inclusion studies. They are focused on small inclusions of fluids and vapor in the minerals, and these fluids are trapped during the cooling of the minerals, during the growth of the minerals, during tectonic phases and fissure development. These, so we can analyze by cooling and heating the temperatures where the fluids were trapped. But so this seems to be a perfect method. We can exactly analyze the fluid temperatures, but these fluids were trapped under certain PT conditions, pressure temperature conditions in the subsurface. Therefore, if we want to use the temperatures we get from our analysis here on the surface, we have to do a correction for the calculation of exact paleo temperatures regarding the original PT conditions. This is very often not very clearly to do. There are certain uncertainties in the estimation of the PT conditions and the fluid chemistry, which again also leads to uncertainties in the paleo temperature calculations. And finally, something that is applied in relatively many studies in modern times, thermochronology, which is based on fission tracks analysis in apatite and zircon, or sometimes combined with apatite, zircon, uranium to thorium, helium analysis. These fissions are formed by the decay of uranium-238 in the crystals, and due to the length of the fissions, we get an indication on the paleo temperature and the time since these minerals have left the annealing temperature. Here we see an example, and we see these boxes over here. And the whole time temperature modeling in thermochronology is based on detailed depositional history and paleothermal data that have to get included into the modeling. So we need a certain set of data to get into the model system into the modeling software for the thermochronological analysis. These are the boxes. These are the data that we have from other methods, depositional history, maximum thermal overprint, and then we can create a relatively accurate, very good time temperature modeling of cooling, heating, cooling, heating. But without this detailed depositional history and also additional paleothermal data, thermochronology will be something like here, these pale colors, which are badly constrained. We have wide arrow fields and are not very precise. So in a way, thermochronology can be done as a tool for time temperature modeling if we have already qualified maturation parameters, but it's not really a primary maturation tool to work with. These are the theoretical backgrounds, and so we have looked on the Vitrinite reflectance, on the benefits and limitations. We have looked on the major other maturation parameters, on the benefits, on how it works, on the benefits, on the limitations. And now I want to sum up these theoretical background in 3 case studies and show you how this works together in these different projects. So the first case study is from the Lower Carboniferous in northern Germany, and it's a classical hydrocarbon province. Northern Germany is one of the oldest hydrocarbon provinces in the country, and it's well known. It's a lot of studies, lots of wells. So we have quite a big database on the geology, on the basin development, on the maturation in the North German basin. And this part here is a part of the maturation map from the North German basin. So the North German basin goes up here to the north. North Sea would be somewhere over here. And here we have the southern margin of the North German basin. And you see here these reddish and yellowish colors are gas to over maturation, while the greenish colors over here are towards oil window maturation. The major targets in this study were the organic rich carboniferous shales, black shales, which are not really black shales. They don't have the high TOC values black shales should have. TOC is mainly below 1 to 3%, sometimes up to 5%. And what is obvious is a very poor kerogen quality. You see all these samples are plotting down here, extremely low hydrogen indices and very low oxygen indices also. The maturation in this area, so the study area is in this blue frame. The maturation seems to be very high. So we have something from about 2% to over 4%. Here we have even areas with up to 5% vitrinite reflectance. So this means maturation is in the dry gas, upper dry gas to over mature interval. And the vitrinite reflectance decreases to the north towards the basin center. That's something we see here, which is somehow strange because normally maturation increases towards basin centers and not decreases. So the idea on this setting is that we have the very poor hydrocarbon potential, as we see here in these samples, due to intense hydrocarbon generation during high maturity. So due to the high maturity, all organic matter that could be transformed is transformed. So we have been going almost through the entire gas interval, produced all the gas that is possible, and what is left over, the residual kerogen, is seen here. So this very poor HI, OI values are a result of the residual kerogen. So if this interpretation is right, this would be a highly prospective gas play, shale gas play here in this high mature area. And that's the reason why this was a relatively big shale gas concession from one hydrocarbon company. Another company that was interested in or was scanning all over Europe regarding shale play potential, particular shale gas potential, asked me for a new study because they really wanted to know, is this the right scenario or is there something wrong with it? And so we sampled all the different, a set of outcrops, shallow wells from these black shale intervals, the lower interval here directly at the base of the Lower Carboniferous, at the base of the Visean, a second black shale interval, and then at the base of the Lemurian, a black shale interval. And most of these samples, so I isolated the kerogen from the rocks, did slides and had a first look on the slides before I started to do the blocks for the Vitrinite reflectance. And most samples are strongly dominated by dark, highly dark, or mostly black, highly mature keratin. These are all these black, grayish scale. All this is highly mature, very dark to black organic matter. And you see this is strongly dominating, and this fits perfect to the idea of high maturation. But in almost all samples, also less mature keratin is present. And these are these brownish colors over here. One sample with a lot of this material over here. So all these brownish colors indicate material that is not that dark colored like the rest, and is surely not on this high mature level like this dark stuff over here. It's mostly only very low amount, a few percent, as you can see here, mostly below 10%, often below 5%. But this little bit of organic matter questions the high maturity for the southern North German basin. Because if this southern North German basin was heated up to high upper gas to over mature conditions, all organic matter in the system has to be cooked. So if I take a pot and put it on the oven and cook it, everything in the pot will be cooked. There is nothing left uncooked. So this is a big question mark and had to be solved. Here are the slides that I checked, and here we see the different scenarios, and you see these examples that you have seen before. This is a slide where we have lots of black, very dark to black organic matter, high mature, but also some brownish material over here. And here, this is the one sample where we have lots of brownish material and very little organic matter. All these particles are here, brownish material, which are slightly less mature than this black stuff over here and over here. And what we see in vitrinite reflectance is the same. So we get vitrinite, high mature. The huge majority here in this sample is high mature from 1.6 to over 3%. But we have a small population here, which is much lower in maturation. And the same here, we have this low maturation part of the assemblage, and we have the high maturation part again, up from 1.6 to almost 3%. And in detail, we see here the total vitrinite reflectance, 2% here, 1.2% here, but the in situ maturation, which is really relevant for the identification of the basin thermal history, is relatively similar, 1.18 to 1.03. So we were looking for more samples with relatively well-preserved vitrinite to get reliable data from vitrinite reflectance, and we ended up with a number of samples from different outcrops. So in total, this study was based on 50 samples. You see here we worked with 2, 4, 6, 8, 9 samples. So there was not such a high amount of samples that really had well-preserved vitrinite, but we decided only to take only the samples with really well-preserved vitrinite, because this really gives reliable data. We could take all 50 samples, but then the high majority would give us some reflectance data which are not really reliable. So what we see here is that we have in the total assemblage, we have a widespread from almost 1% to almost 2.5%. But if you look on the in situ assemblage, we see a relatively narrow range of maturation from about 1% again to almost 1.2%. Only this one sample over here is a bit higher, 1.43%, which seems to be related to the structural tectonical setting this well is placed in. And again, the reworked assemblage has again a relatively high spread from maturation between 1.4 to 2.5. So what we see here is that the in situ maturation is relatively similar in all the samples in this area, in this study area, and it's much lower than the total maturation and much lower what we have seen in the map before. So if we plot this into this map, so here we are in this high mature area in the southern North German basin, but all the maturations, all the vitrinite reflectance falls in this interval between 1 and 1.5%. So the upper oil window to wet gas, and not in the upper gas to over mature, as it is shown here in this map. This makes a major change to the maturation, to the interpretation of the maturation of this basin, and of course to the evaluation and the potential for shale gas in this area. If we are only in the upper oil to mixed zone, then there is no potential for shale gas, and then there is no reason to hold this license any longer. And so we see here that we have a similar maturation as it is common in the North German basin. So this makes things also more normal, because now the southern margin of this basin is no longer so hot compared to the basin center. But why do we have these high maturation always mapped here in this area? This is due to the high amounts of reworked organic matter from the active Variscan origin south of the area. So here we have so this is part of the Variscan origin. And this Variscan origin comes from the south, prograding towards the north in the final Variscan orogeny, and brings a lot of sediment eroded from the Variscan origin into the basin over here. And this is very often highly mature material included into this orogenic wedge, heated up, metamorphized partially, and then eroded into the basin again. And so, due to the high amount of eroded, recycled sediments, we also get a very high amount of high mature recycled organic matter in this interval, and that's the reason for these high maturations in this southern margin of the North German basin. This is just by the high amount of high mature reworked organic matter, reworked vitrinite. So far to this study, so it shows that vitrinite reflectance was done here on quite a number of samples, but can be very misleading if you do not realize vitrinite recycling and also not realize vitrinite preservation. But these are really important parameters that has to be taken in account when you analyze vitrinite reflectance and interpret your vitrinite reflectance data. A second case study.
Excuse me, just to let you know, you've been going for about 90 minutes now, okay?
Okay. Okay. I will come, I'm close to the end. I will just show two more examples on this vitrinite reflectance. Here, from the Paleozoic in the Cantabrian Mountains, again a Paleozoic example. What we had was chemical analysis, Tmax data, vitrinite reflectance calculated from Tmax data, and the production index, and they showed some very erratic distributions. These are not two sections, but two formations sampled in different sections. And we see a quite big spread here in the Tmax and also in this calculated vitrinite reflectance, which is not available here in the production index, showing again this misfit between production index and other maturation data. But we got more data. conodont alteration index data and also fluid inclusions data. And the conodont alteration index data shows a very homogeneous maturation for this interval over here. Very low maturation in this interval, higher medium maturation in this interval, and slightly lower maturation down here, which is not really fitting to the spread that we see in the Tmax and calculated vitrinite data. And also here we see fluid inclusions data, which show a relatively minor changes between 110 and 140 centigrades, but do not fit to the variations in the Tmax data again. And in the area we have also clay mineral data, which are showing very high maturation, middle to upper gas window maturation. So we see we have a handful of different maturation parameters, and that don't work together. They are all indicating different things. So there's a strong inconsistency between the different maturation data sets. How can we handle this? How can we understand this? So this was a point when I get asked to do vitrinite reflectance in these samples. And these are the vitrinite reflectance data from the measured samples. And we see that and this is based on the in situ vitrinite. Only the analysis of the in situ vitrinite. All these samples had two populations, and we just only interpreted the in situ population, and this gives us a very narrow range in the lower to middle oil window for all the samples. And we see no longer any local anomalies as in these Tmax data. The partially increased conodont alteration and fluid inclusion data are related to hydrothermal fluid systems in the carbonate formations between the clastic formations where we took these vitrinite samples out, which is also indicated by hydrothermal dolomitization in these carbonates. And the difference between Tmax, this is spread in the Tmax, and the relatively narrow range in the vitrinite reflectance, is just due to the different amount of highly mature recycled vitrinite. Again, here we have this effect of mixing high mature with low mature. The unusually low vitrinite reflectance value in the lower formation, this value over here, is caused by massive alteration of vitrinite. As I said before, if we have poorly preserved vitrinite, we get lower maturation, lower reflectance, and this is the case here. The whole sample is strongly degraded, so we get a less lower reflectance than the other values. The high maturation from the clay minerals from the Kubler index are interpreted as a result of clay mineral recycling, analog to vitrinite recycling, at least partially. So in this study, the vitrinite analysis, vitrinite reflectance analysis, could be used as a base for correlation and interpretation of the multiple maturation parameters, because with the vitrinite reflectance, we realized the anomalies in these data set here are just related due to mixes of high mature versus low mature material, not to real changes in the maturation environment. And the final case study from the Paleozoic, again in Northern Africa. Again, we have a data set of maturation data, vitrinite reflectance, also again measured vitrinite reflectance, thermal alteration index, Tmax from Rocky-Val, and production index. And we see that all these different data sets do not really fit together. In the vitrinite reflectance, we see very low maturation in this upper part, slightly higher maturation in this part here. The TA shows us pretty high maturation already here in this upper part, going into the upper gas window over here. The Tmax again goes all along in the base of the oil window, and also the production index is very low, except of this lower part here. Very interesting is that the magmatic intrusions, these yellowish lines here, seem to have no impact on the maturation. So in no parameter we can see any clear effect on the maturation levels by these magmatic intrusions. And this is a more than 100 meter thick magmatic duct, but without any effect. Also, this area is a well-known gas play. So if we are not even getting into the gas window in all these parameters, what is the source for the gas? In our study, we first did a high-resolution spore color index analysis, which is shown here. So we have this upper part here still in the upper oil window, and then we get a little jump on a major unconformity at the top of the Paleozoic, and we get into the mixed zone and into the upper gas window here. And the spore color index shows a clear relation to the magmatic intrusions, what you would expect, because we have very high heat flows in the direct areas around these dikes, so this should have an effect on organic maturation. To back up these spore color indices, because color indices are very often not really taken serious in industry, and very often taken very doubtful. So we took some samples for vitrinite reflectance, which are here, the red ones, which show the very similar trends to these compared to the spore color index. So this changes the whole scenario from a very low mature to a high mature setting on this full lower interval over here. And the magmatic intrusions show a major effect on maturation of the organic matter, as we should expect. So based on these data, we see a two-phase maturation history in this area, and particularly in this well, and also a two-phased hydrocarbon generation. The primary maturation, these blue lines over here, represents a general maturation trend, which is a basin-wide maturation related to the Paleozoic basin burial. So this is something that you see in all wells in this area. This is only one well from a big set of wells that we worked in. And so from this Paleozoic burial maturation, we get these lower hot shale down here, the lower source area, into the upper gas window, and get this productive to produce the gas that we find in the area. The upper hot shale, which is up here, will be in the lower to middle gas window based on this basin maturation trend. The secondary maturation is a thermal overprint by the Mesozoic magmatic intrusions. So this is much younger, and this has no change in basin-wide general maturation, but has a massive effect locally. So we see here in these areas where we have the magmatic intrusions, we get a huge change in maturation. So this red part is the effect of the secondary heating by these magmatic dikes. This leads to additional gas generation, at least in this well, because the set of dikes here gets into this upper hot shale interval and shifts up the maturation in this organic rich shale interval into the upper gas window, and leads to a massive production of gas in this interval. That's the reason why we have both hot shale intervals activated in this well, and we have in this well two gas plays above each other, one down here and one up here. The one sourced by the lower shale interval, and this one up here by the upper shale interval. And we see in this study also that the swarms of thin dikes are much more effective on maturation, heating up this whole big interval, than very thick isolated dikes, as we see this one here, which has a relatively fast downturn in maturation again away from the dike. So again, with careful detailed organic matter analysis and vitrinite reflectance analysis, this complex story could be deciphered in very detail. So coming to the end, what you should take home with you, there is no perfect maturation parameter. I'm very sorry for this, but all parameters that we use for maturation have the benefits, but also have the limitations. Sediment and kerogen recycling and alteration is a crucial issue of maturation analysis. And that is something that I feel is very often underestimated in maturation studies. All chemical parameters are based on bulk rock analysis. Therefore, kerogen recycling, preservation, and other processes like this cannot be recognized. Chemical parameters not only controlled by maturation, but also by depositional setting, primary composition, diagenous alteration of rocks, and kerogen, as I showed you in the different methods before. The reflectance of non-vitrinite organic particles and the use of the so-called vitrinite reflectance equivalence is not really developed yet. It needs much more studies to become a really solid, robust maturation parameter, and it's always calibrated versus vitrinite reflectance, so it can't be better than this. Vitrinite reflectance, if it's performed carefully and corrected for recycled and altered or caved vitrinite, delivers highly accurate and reliable maturation data. So coming back to the question from the beginning, is there anything better than vitrinite reflectance in organic maturation? In my opinion, no. There are other maturation parameters that might be as good as vitrinite reflectance, but there's surely nothing better than vitrinite reflectance. And if it's done carefully and performed in a good way, vitrinite reflectance is still one of the best maturation parameters that we have in organic maturation. And with this, I will finish my presentation and will point over to you for question and discussion.
All right, thank you, Hartmut. It was nice these case studies in the end. Always good to see some data. Is there any question that would come from the audience now? Yeah, maybe I was just, when you were discussing these I mean, the way you perform the analysis of vitrinite and how it evolves with temperature, I was thinking back about the talk that was given on the models that describe maturation of vitrinite reflectance. And how in recent years there was this basin RO model, and then recently Burnham's improved model, easy RO dogleg. Do you have any comment on it? Do you see any of these behaviors with the change in the trend of maturation with temperature? Do you see it in the data anywhere that you worked?
I had this discussion several times with people, and sometimes they say, okay, if it's so different what you present here, so all these correlations, all these models are more or less wrong and useless because they are built on the old data, on data like from the North German Basin, for example, which are obviously not correct. I guess it's not that bad that we have to create totally new models, but maybe we have to confine some models, and maybe the problems with some of the models that we have up to date could be solved with more precise vitrinite reflectance analysis, as I showed it in this way. If you remember the presentation at the EAGE meeting in Paris, where there was the new equation and a new method used to calculate maturation from Tmax, and also generally the correlation from Tmax to Vitrinite reflectance, these different equations from Sweeney and Burnham and others, I guess that they don't fit perfectly between the modeled and the real data might be not a problem by the algorithms or by the equations. It might be more a problem by precise Vitrinite reflectance analysis. So when I saw this talk in Paris, I thought, okay, there are some data that don't fit, but maybe we just have to reanalyze these samples if we can get better Vitrinite reflectance. And then if we have better Vitrinite reflectance, we might maybe able to fine-tune the equations even better. I guess that's a problem. It's not that it doesn't work, that it makes no sense to have these equations, have these models, but I guess the misfits that we still have between the models and the measured data might be not only related to mathematical problems with the equations and models, but also with measurement problems on the Vitrinite side.
And related to that, so in recent years, I came across the idea that maybe there are different types of vitrinite from different plants, or maybe different periods of time, that mature in a different way, and maybe require different models. And maybe part of it may be related to the content of sulfur, but part of it may be the actual type of the material,
yeah.
or environment of the source rock, or the particular kerogen assemblage around it that may control the way it matures. Can you comment on
that? Yes, I guess that's a point that I will also focus on in the webinar next month when we talk about kerogen composition, because that's a mixture of more detailed analysis of the kerogen composition and the maturation. So that's also quite often you find comments on different maturation when different kerogen types start to generate oil or gas, for example, type 2, type 3. You very often see there are certain differences in initial temperature to start with. And I guess that's a point. If you have different types of organic matter and also different types of chemical background, sedimentary phases, for example, sulfur-rich sediments and so, these processes start relatively early, but or earlier than in the normal case. But again, this would really need to have very detailed maturation analysis and compositional analysis of the kerogen to understand what is really controlling these differences. Is this really based on compositional effects? Is this maturation that it mainly controls it? Is it something time dependent? There are also models in hydrocarbon system analysis which are differing between Paleozoic systems and Mesozoic systems. The Cetaver stuff, for example, they have the one kerogen type for Mesozoic, Cenozoic materials, one for Paleozoic materials. I think that's not the really problem. It's not so much about age. It's much more about composition of the organic matter. And we have, in most Paleozoic samples, we have in the shelf environments a different composition than in Mesozoic or Cenozoic times. I guess that's a major problem. It's not related to timing because it's older or younger. It's just related to the changes in the kerogen composition. I can't hear you. Can you hear me?
I'm sorry, I forgot to turn on my microphone. So it looks like we are getting onto the same issue like with other processes and models, that basically turn out to be dependent on multiple parameters, not just temperature or pressure. So I see that this was nicely solved for quartz cement precipitation with the work of Geocosm company and people around it, and Walderhaug initially in the 90s. But for the other processes such as Vitrinite or maybe some of these other thermal indicators, we haven't solved this yet. For example, when you mentioned the indicator via smectite illite, in my opinion, this essentially is totally uncalibrated tool that is completely useless, because there are so many variations in the way smectite illite goes on, because it's not just a function of the burial, but it's function of the composition of the fluids that are interacting with the recrystallization. So in my opinion, smectite illite indicator is just completely useless to any maturation indication.
Yes, I would more or less agree with you, but it's used again and again. That's the reason why I just put it in. And the problem is that all these clay mineral stuff is, of course, not only controlled by temperature, by maturation. It's a lot, maybe much more controlled by chemical changes, chemistry, diagenetic alteration. So if you have diagenesis, we have changes in the chemical composition of the sediments, and this will affect clay mineralogy quite a lot. I guess much more than temperature, and therefore these clay data, which are sometimes taken as very reliable, serious maturation data, in my opinion, has to be taken, very, very careful. And would be, in my opinion, surely no backbone to any maturation interpretation. And also they like crystallinity, because that's what we realized in several studies, that we had, based on the organic matter, relatively low maturation, but a relatively high illite crystallinity. And this only can be an effect of reworking, of recycling. So it's not only organic matter or quartz grains that get recycled in the sediment, it's also clay fraction that gets recycled. If I erode clay stones or shales, this material will end up in a new sediment and will give the high matured signature from the primary precursor sediment into the younger one. And that's a point that I realized very often in maturation studies. Doesn't matter what parameter you use, is that, as I said before, the effect of recycling and alteration is strongly underestimated. It's when we talk about sediments, when we talk about reservoir properties, for example, diagenesis and so on, this is all understood. We have recycled sediments, we have grown minerals and so on. But if you talk about maturation, a lot of people think, okay, this material that we have in our rocks that we analyze for our maturation interpretation, this is just in there and just gives us the thermal history of the basin. But also in these parts, we have a lot of recycling and alteration, and particular clay minerals are very, very easy to alter in changing environments during diagenesis.
The first question that I see here is from Anna Hoffman, and Hartmut, you can read it yourself, but for the benefit of the archive later, I will read it aloud. I have a few questions. Is the difference between reflectance minimum and maximum negligible for samples with the maturity in the oil window or below? And do you have any comments on fluorescence alteration in multiple macerals and combined inertinite vitrinite reflection? And how do you handle vitrinite reflectance suppression, for example, in hydrogen-rich vitrinite?
Okay, these are quite some questions to handle. So starting at the top, the difference between Rmin and Rmax in the oil window is relatively small, and it's more or less in the range of the standard deviation of our analyzed data. So in a way, at least in the lower part of the oil window, it's more or less negligible, and towards the upper part, there it might make a difference. The problem is, I don't, even if it's very similar, I don't like to use these terms if I just work on a statistical procedure as for the dispersed kerogen, because it indicates that we really have maximum or minimum data, which we don't have. So even if it's not a big difference, I would clearly use the different terminology to make sure what type of data we have. If you get higher up into the mixed zone and into the gas zone, the differences really become more and more significant and cannot be ignored any longer. So for the lower to peak oil window, they are pretty small. The next question, fluorescence alteration in multiple materials. Fluorescence, yes, fluorescence is a very interesting topic. I had lots of discussions with different people. The first point is fluorescence is controlled by maturation. All different types of organic matter or certain types of organic matter show fluorescence, not all. And this most of the organic matter that would be classified into kerogen type 1 and 2 show fluorescence. The terrestrial plant debris like kerogen type 3 and 4 does not show fluorescence at all. And that's an issue that will be dealt with in the next webinar in October when we talk about kerogen composition and optical analytical methods to identify kerogen composition and kerogen quality. But that's a point. So, but if we have material that shows fluorescence, the fluorescence stays up to the end of the oil window. Around 1.3 to 4 percentage of vitrinite reflectance, the fluorescence is gone. And even if you have strongly fluorescing or originally strongly fluorescent minerals, all the fluorescence is gone. And there is a discussion. Some people don't believe that alteration, so degradation of organic matter, don't affect fluorescence. So from my observations in all the different studies I did, working with very often not very well preserved material, fluorescence is the same in a well-preserved organic component as in a totally rotten organic component. It's not so intense any longer because all the parts of the organic matter that are decomposed and are rotten don't show any fluorescence, but the well organic material that is remained in these rotten flocks still shows the same fluorescence as well-preserved particles. So that's a reason why fluorescence is very, very helpful to identify the origin of amorphous organic matter in many studies. So what do we have next? How do you handle vitrinite reflectance suppression, e.g. in hydrogen-rich vitrinites? The point is that's a problem, and this is something that I will focus more in the next talk about the concept of kerogen concept we work with. So if we take very precisely from the initial definitions, the kerogen types 2, 3, then we have no hydrogen-rich kerogen 3. The hydrogen ratios from type 3 kerogen, as described initially in the initial papers, is pretty low. The problem is in the geochemical way, we very often have this mix of type 2, sometimes type 1, 2, 3 kerogen organic matter mixed in one sample, And then we, depending if it plots further to the 3 or 2 graph, we call it 2 or 3 or mixed 2, 3. And then we have hydrogen-rich stuff and hydrogen non-rich stuff. But vitrinites, the cellulosic material in itself, as I showed before, this is cellulosic material. And you can Google even and look for cellulosic material. Cellulosic material is never hydrogen-rich. And that's a point, we have no hydrogen-rich vitrinites. If you have hydrogen-rich samples where we have vitrinites from, the hydrogen-rich signal is not in the vitrinites, but in the other parts of the kerogen. And that's the reason why I cannot really answer this question, because working with an optical-based concept of kerogen classification is pretty much different from this geochemical-based approach. But as I said before, this will be part of the next webinar when we talk about kerogen classification, kerogen composition and interpretation. That's something I left completely out of this webinar, because here today we were talking just about maturation. I hope this answers at least Anna's question. So Wayne came up with something more. I have a new question from Paul. Wait a minute. With these low readings. Yeah, to Wayne's question, did you observe material that produced these low readings? That's a really problem. I haven't seen the original slides, which are the base for this initial studies on the vitrinite reflectance in Northern Africa with these very low values. In my slides, I have a certain amount of poorly preserved vitrinite, which could lead to very low reflectance numbers. But there are also quite a lot of well-preserved vitrinites, so I'm not sure if they just picked out the poorly preserved stuff to get these low numbers, or if there's any other reason. It's also a point of processing the samples and doing the slides. So before I came into this project in Northern Africa, I get asked by the company that we worked for if it would be possible to do palynology and organic maturation with this material, because they got slides from another company, from another consultant, which were absolutely poor. They showed me some slides, and there was nothing in these slides that was really worth to work with. So I got the original samples, and we did samples in our lab, and we got a lot of organic material. We get well-preserved stuff, phytoclasts, spores, chitinozones, architects, all the stuff that you should get in these environments. So we could do a very good palynostratigraphic and palynological study, and also maturation study. So I'm not quite sure how these very low numbers come together. This is a mix of bad processing and taking the wrong particles. I'm not quite sure. I do not really understand why they have these extremely low vitrinite reflectance numbers, and I will never understand until I get the original slides, but they were not to get so far. And now this project is off for almost 8 years, so I will never go back to this.
And then Paul Comet asked, surely Rock R0 solves most of these problems.
Yeah, also the other question, decellulose during maturation. Yes, it is. With increasing maturation, we get a sort of decomposition of the original material, both cellulosic and lignite material, and that's the reason why we get different structures and different reflectance. So, but the initial material, and that's why I was talking about the hydrogen-rich vitrinites, the initial material is cellulosic material that we start with and lignin, and both are not hydrogen-rich. So during the maturation process, they get decomposed, and you can call it decellulosed or whatever you want. And the other part, vitrin, is just lignin in a different states of demethoxylation. Vitrinite is both, is cellulose and lignin material. And it's mainly, from my understanding, cellulose material and not lignin. Lignin are only these very, very narrow laminae in between the cells, which are mainly made of cellulose.
Yeah, Jürgen is asking, Hartmut, as it
question.
seems to be very crucial to determine between in situ and or recycled vitrinite, how can you be always sure in determining the origin?
That's a good question, and that's not easy to answer. In the case studies I showed you, it was quite clear because there was no caving. We didn't work with valves, with cuttings, or with cuttings. We worked with valves, real valve samples taken from the cores, and also outcrop samples in Spain and also in Germany. It was mainly outcrop samples with a few core samples, and in Northern Africa, it was mainly core samples. So there was no problem of caving. If we have cuttings, we get several populations. We might get a caving population, which generally is less mature. We get an in situ population, we get a recycled population, and we can also get several recycled populations, as I said before. The sample, the most extreme sample I have seen so far included 6 different populations of recycled vitrinite from about 1.7 to over 5%. And they were all clearly separated, clearly to identify, and were also to identify in several samples out of this section we work in. So this is not always that simple, and if we work with non-caved material, the lowest maturation has to be the in situ maturation. Because as I said before, if you take a pot and put it on the oven and boil it, so the temperature that you boiled is found in all material in the pot. So the lowermost material, the lowermost maturation in your mix indicates the maturation where the current depositional system was phase 2. All the higher maturations, only seen in parts of the kerogen, cannot be from this environment, because then it should be seen in a similar range in all of the organic matter. So the problem is when we have cuttings and we get the option of caving, because then we can also have low mature populations from caving, and then it gets tricky. And that's very often because why I like to combine color indices, polynomorph color indices, and vitrinite reflectance in maturation studies, in integrated maturation studies, because then I can check from my slides for the polynomorph colors what type of polynomorph I have in the different maturation populations, and can understand what is caved, what is in situ, what is recycled. But it's no easy solution to this problem.
And Wayne is asking another question. I was pleased to hear that you have observed multi-populations of reworked vitrinite in samples. I've seen this on several occasions as well. Have you observed a reverse maturity trend with depth over a large interval if you plot the reworked data?
Slightly, yes. It's sometimes, very rarely I've seen this. I guess this is, if you have a very simple setting where you have something coming up, like in this Virgin origin, this origin comes up and you get a sort of gradational erosion during the different maturation levels, you can see these clear trends. But if you have an hinterland where you have totally different areas of high and low mature environments, which are all come together in sedimentary systems, and most of the shelf sediments that we normally work in for hydrocarbon exploration are derived from huge areas drained by big river systems. So we very often get a sort of mix from all different maturation settings in the hinterland in this samples on the shelf. So I very rarely find these reverse trends, and not very clearly, but always pretty weak.
Nazim Looney is asking, extending the VR solid calibration robustness to non-terrestrial particles, algae mainly, and to start with maybe, seems to be most useful in future of maturity analysis in vitrinite poor samples.
Yes, I fully agree to this. And that's something we did in studies in the Arabian Peninsula, where we are working mainly in early Paleozoic shale systems, where we don't have vitrinite, where we have huge problems with precise maturation analysis. And we started to develop these non-terrestrial particles using graptolites, using chitinozones, using amorphous material, using architects and cryptophores. The problem is, so we get quite some initial data, but also there we have only a limited maturation range. And what I would like is to do a study over a wide range of maturation where we can calibrate these different types of organic matter to each other. Because we developed now with this project on the Arabian Peninsula very good technical tools to clearly identify the different types of organic matter, which, as I said, is not so easy on the polished surface. It's a bit more time consuming, but we do special types of resin blocks where we can focus into the resin block and to see what type of organic matter really is behind this polished surface, because on the polished surface, very often amorphous material, chitinozones, graptolites, and even sometimes scolectrons look very, very similar. But with this method, we can clearly identify the different types, and we can get very precise reflectance trends for each group. But what we need is more maturation environments. So there is a big need, in my opinion, for non-vitrinite reflectance tools, but we need good studies, long-ranging maturation studies to develop these tools and to calibrate these tools before we can really start to use these tools. But that's definitely a very strong point for the future maturation analysis.
Mm-hmm. Okay, I don't see any other questions coming in. So thank you everyone for your attendance. And we'll see each other again next month for the continuing talk, again given by Hartmut. Right, thanks everybody, and goodbye, have a good evening or day.
Thank you very much.